Welcome back to the deep dive. We're here to break down complex sources and give you those crucial insights. Today we are tackling something really tough. Leptomeningeal metastasis. LM for short. It's one of the most aggressive ways advanced cancer can progress.
Yeah, it's truly devastating. LM means cancer cells, often from places like the breast, lung, or from melanoma, have basically broken away and invaded the central nervous system. They get into the leptomeninges, those delicate membranes surrounding the brain and spinal cord, and specifically into the cerebrospinal fluid, the CSF, that fills that space.
And this is where it gets really strange, based on the research you sent over. When this invasion happens, the body seems to, well, freak out. Clinically, you see this massive inflammatory response in the patient's CSF. Loads of immune cells rush in. It apparently looks a lot like infectious meningitis.
Exactly. You've got immune cells everywhere. They're activated. They're present in huge numbers. It looks like a fight is happening.
Yeah.
And yet, the cancer cells just keep growing. They thrive. And that's why LM has such a grim prognosis, historically.
So that's the core puzzle, right? Why does the immune system, even when it's so obviously revved up, fail to stop the cancer in this specific spot? What is going on?
Well, the answer they found is pretty surprising. And it really centers on one specific molecule, a signaling protein called Interferon-gamma, or IFNγ.
Interferon-gamma, okay.
Yeah, using really detailed analysis of patient samples, they saw the CSF was just packed with inflammatory cells and, importantly, rich with IFNγ and signs of its activity. But here's the kicker, the bit that kind of flipped the script. Finding higher levels of IFNγ in the CSF at diagnosis actually correlated with patients surviving longer.
Wait, really? So more inflammation, more IFNγ is actually good?
In this context, yes.
Sure.
It suggested the immune system wasn't totally failing. It was deploying an anti-tumor signal, IFNγ, but maybe that signal wasn't quite strong enough, or perhaps it wasn't being interpreted correctly by the other cells involved.
Okay, so our mission for this deep dive is clear. We need to unpack this unique pathway. How does IFNγ work specifically in the leptomeninges to suppress cancer? And we need to identify the key players, right? You mentioned 3 roles. The cell giving the go signal, the orchestrator cell, and the executioner cell that actually takes out the cancer.
That's the plan, unraveling that specific chain of command.
All right, let's start at the beginning, the go signal. If IFNγ is so critical, who's actually making it there, right in the thick of things in the leptomeninges?
Good question. The researchers used techniques like flow cytometry and single-cell analysis, looking at both human CSF and different mouse models of LM lung, breast, melanoma cancers spreading to the leptomeninges. And they quickly zeroed in on 2 usual suspects, 2 types of immune cells known for packing a punch: T cells and natural killer cells, or NK cells. Both were definitely producing IFNγs.
Okay, T cells and NK cells. Are they both equally important here? Is it like a tag team effort?
You'd think so, perhaps. But surprisingly, no. They did these clever experiments, selectively getting rid of IFNγ production in one cell type or the other. And while NK cells do make IFNγ, it turned out that the T cells were responsible for the majority of the IFNγ BS that actually had a measurable anti-tumor effect.
So taking out the NK contribution didn't really hurt the overall defense much?
Not significantly, no. The protective effect, the slowing of tumor growth, was largely still there. So in this very specific environment, the T cell is the primary generator of that crucial IFNγ signal.
Okay, T cell generates the go signal, but a signal's no good if no one can hear it. The host, the body itself, needs to be able to receive that message, right?
Precisely. And they confirmed that too. They used mice that were genetically engineered to lack the receptor for IFNγ, it's called the IFGR1. In those mice, without the ability to hear the IFNγ signal, the leptomeningeal tumors just grew completely out of control, wildfire.
Wow. Okay, so it proves the host response to the IFNγ is absolutely essential.
Undeniably. Without that receptor, the whole anti-tumor program collapses.
This brings up a really key point then. Is the IFNγ acting like a direct weapon against the cancer cells, or is it more like telling other cells in the environment what to do?
Great question, and they tested that directly. They took the cancer cells, the leptomem cells, and specifically knocked out their ability to respond to IFNγ by deleting their receptor subunit, IFECR2.
So if IFNγ kills directly, these modified cancer cells should now survive and grow faster.
Exactly, that was the hypothesis. But their growth rate didn't change at all.
Get out, really? So the cancer cell doesn't even need to hear the IFNγ for the immune system's plan to work?
That was the stunning part, yes. It means IFNO controls the cancer growth entirely by influencing the surrounding cells, the immune cells, maybe other cells in the microenvironment, not by directly targeting or killing the tumor cell itself.
It's an indirect hit. The signal marshals the host's defenses.
Correct. It's all about orchestrating the host response.
Okay, this is huge because so much of modern cancer immunotherapy, things like checkpoint inhibitors, relies on the adaptive immune system, right? On T cells recognizing specific flags, antigens on the cancer cells. Does that whole process, antigen presentation and T cell recognition, even matter here in the leptomeninges, given what you've just said?
Well, conventional thinking says it should. But this environment, the CNS, is often unconventional. So they tested it. First, they showed that just artificially boosting IFN levels locally using a safe viral delivery system called an AAV could control the tumor growth in LM. That worked.
Okay, so boosting the signal helps.
Yes. Then came the critical test. They took that same AAV IFNO system and put it into animals that completely lacked adaptive immunity. These were RAG1 deficient animals, no functional T cells, no B cells, no way to do that specific antigen recognition and presentation we associate with adaptive immunity.
So basically asking, can the innate immune system, the body's first responders, handle this job alone if you just give them enough IFN signal?
Exactly that. And the answer was yes. The anti-tumor effect, the tumor control driven by IFNO, was completely preserved. It worked just as well in animals with no adaptive immune system at all.
That is fundamental. It means the whole mechanism fighting LM is distinct. It doesn't rely on the T cells seeing the cancer in the traditional way.
It's a profound difference. It operates independently of adaptive immunity and antigen presentation. This specific location dictates a different set of rules.
Wow. Okay, this feels like a good point to pause and just quickly recap what we've learned before we hunt for the orchestrator cell. This is complex stuff.
Good idea. So key takeaways so far. First, the paradox. LM triggers intense inflammation in the CSF. You see lots of immune cells, but the cancer grows anyway.
Right.
Second, the generator. T cells, specifically, are the main source of the critical anti-tumor signal, Interferon-gamma or IFNO.
Got it. T cells make the signal.
Third, the non-target. That IFNO signal doesn't work by hitting the cancer cells directly. Its effect is entirely on the surrounding host cells, the microenvironment.
Indirect hit. Check.
And fourth, the immunity type. This whole protective process is driven by the innate immune system. It doesn't need the adaptive system, T cells recognizing antigens, B cells to work.
Okay. So, T cells make the signal, it works via the host environment, and it's an innate process. Which leads us directly to the next question. If adaptive immunity isn't required, which innate immune cell receives that IFNS signal from the T cells and actually organizes the defense, who is the essential orchestrator?
Right, so the hunt was on for the innate responder. They looked at which innate cells showed signs of responding to IFNS, technically. They looked for phosphorylation of a protein called STAT1, which happens when the IFNO receptor is activated. This pointed towards several candidates: monocytes, macrophages, neutrophils, and also dendritic cells, or GCs.
Okay, a few suspects. Did they rule any out? Give them alibis?
Yes, they did. They started with the more common myeloid cells, monocytes, macrophages, neutrophils. They used genetic tools, specifically something called a lysomic driver, to delete the IFNA receptor only in those cell types.
And what happened when those cells couldn't get the IFNO signal?
Well, the tumor control was fine. Removing the receptor from those cells didn't impair the anti-tumor effect. They were essentially dispensable for this particular job in LM.
Okay, so not the monocytes, macrophages, or neutrophils. That really narrows it down. What about the dendritic cells, the cDCs?
Bingo. Conventional dendritic cells, or cDCs, turned out to be absolutely critical. They used two different methods to basically eliminate cDCs from the picture. One using a ZBTB46 DTR system, another a CLEC9A approach. And when they ablated or removed the cDCs, leptomeningeal tumor growth significantly increased.
Whoa.
And not only that, but removing the cDCs also made the therapeutic IFO treatment, the AAV delivery we talked about, much less effective.
So the DCs are the crucial link. They have to be there, and they have to be able to respond to IFS. They are the orchestrator.
Precisely. They are the essential intermediary receiving the signal from the T cell.
But we know DCs are famous for presenting antigens to T cells, starting that whole adaptive response. We just established that's not what's needed here. So what is IFNO doing to these DCs? If not antigen presentation, then what?
This was the really cool part, the aha moment, about the cell communication. It turns out IFNO is driving the maturation and movement of these DCs. Specifically, the T cell's IFNO signal pushes a subset of DCs, called cDC2s, to mature into a more advanced form, migratory CCR7 positive DCs. Think of IFNγ as the signal that tells these cDC2s to grow up, get ready, and move out.
Like a drill sergeant getting them ready for a mission.
Kind of, yeah.
Yeah.
It governs their maturation into these highly capable mobile units.
And if you block that maturation signal, if the DCs don't hear the IFNγ,
They tested that too by deleting the IFNγ receptor, specifically in the cDCs. And just as you'd expect, the tumors grew bigger, and they saw a block these mature CCR7 positive DCs fail to develop properly.
okay, mature mobile DCs, where are they coming from? Are they already in the brain? Or
That was another fascinating detail. These crucial CCR7 positive DCs seem to mostly originate from outside the central nervous system, extracranial sources. They aren't primarily coming from, say, the bone marrow right next door in the skull. They're actively migrating into the leptomeninges to do their job.
wow, okay, so T cells signal IFNγ matures cDC2s into CCR7 plus mobile DCs. These DCs migrate into the trouble zone. But what is their job once they get there, if not activating T cells? What's their final order?
Their role seems to be providing cytotoxic support. They orchestrate the conditions for killing the cancer cells. Looking at the gene expression, the transcriptomics, IFNγ didn't seem to directly slow down cancer cell proliferation or anything like that. But when they used immunofluorescence imaging,
Yeah, what did they see?
they saw significantly increased levels of cleaved caspase activity specifically within the cancer cells, but only when IFNγ was present and presumably activating this whole pathway.
Cleaved caspase, that's the molecular switch for apoptosis, right? Programmed cell death, the cell suicide signal.
Exactly. So the DCs aren't killing directly, but they are essential for triggering that suicide program in the cancer cells. They're setting the stage for the kill.
Which means they must be communicating with the final player, the actual executioner.
It points directly to the innate immune system's premier killers,
Hmm.
natural killer cells, NK cells.
Ah, the NK cells make a comeback. We knew they were there, making some IFNγ, but T cells made more. Now they have a different role.
It seems so. The leptomeninges they found contain NK cells in various states, some naive, some activated, some proliferating. They're present and ready.
So how does the orchestrator DC talk to the executioner NK cell? What's the message?
It's all about cytokines, those chemical messengers. The newly matured CCR7 positive DCs, the ones activated by IFNγ, start pumping out specific cytokines. Interleukin-12, IL-12, and interleukin-15, IL-15.
IL-12 and IL-15, I know those. They're huge for NK cells, right?
Absolutely. They are well-known, powerful signals that promote NK cell survival, proliferation, making more NK cells, and importantly, their activation and cytotoxic function, their ability to kill target cells. And crucially, the NK cells in the leptomeninges were found to express the right receptors to receive these IL-12 and IL-15 signals. The communication line is open.
Okay, that makes sense mechanistically.
Yeah.
Did they see evidence of this DC to NK communication in actual human LM patients?
They did. They analyzed CSF from human LM patients and found, yes, elevated levels of exactly these NK-supporting cytokines, IL-12, IL-15, and also IL-18, another related one. And functionally, when they took human NK cells and cultured them in CSF from LM-positive patients versus CSF from controls, the NK cells survived better in the LM-positive CSF. The environment was actively supporting them.
The biology holds up in humans. Okay, so one final step needed. Definitive proof that the NK cell is the one actually carrying out the execution order.
Right, the final piece of the puzzle. They went back to their mouse model where they were boosting IFNγ therapeutically with the AAV. This normally controls the tumor and helps the mice survive longer. Then into that system, they introduced an antibody treatment that specifically depletes NK cells, gets rid of them.
And the result?
The entire anti-tumor effect vanished. Depleting the NK cells completely abolished the tumor control and the survival benefit provided by the IFNγ boost.
Game over. If you remove the NK cells, the executioner, the whole IFNγ-driven defense collapses, even if the T cells and DCs are working.
Precisely. It confirms the NK cell is the indispensable final effector in this specific pathway.
Incredible. So let's put it all together. This study has uncovered a really unique three-part innate immune defense against cancer in the CSF. You have T cells providing the initial go signal, the IFNγ.
Yep.
That IFNγ signal acts on conventional dendritic cells, the cDCs, which function as the orchestrator. They mature, specifically the cDC2s becoming CCR7 plus DCs, and then produce IL-12 and IL-15.
Right.
And finally, those cytokines activate the executioner, the natural killer cells, which then induce apoptosis, that programmed cell death, in the cancer cells.
That's the pathway they've mapped out. A T-cell ZDC NK cell axis, all operating within the innate immune system framework, independent of traditional antigen presentation.
And the clinical implications seem pretty significant then.
Oh, absolutely. It strongly suggests that many standard immunotherapies, particularly checkpoint inhibitors that primarily aim to boost T-cell adaptive responses against cancer antigens, might not be the most effective strategy for LM, because the core essential anti-tumor mechanism here relies on this innate axis involving DCs and NK cells.
So future treatments should probably focus there, on finding ways to boost that DCNK communication loop.
That seems like a much more direct mechanism-based approach for LM. Thinking about therapies that perhaps deliver IL-12 or IL-15 safely to activate NK cells, or strategies to enhance the maturation and function of those crucial cDCs within the leptomeningeal space.
Shifting focus from the adaptive T-cell targets to these innate effectors seems key.
Yes, it opens up new therapeutic avenues tailored to the specific biology of LM.
A really fascinating deep dive into the complexities of CNS immunology and how location really matters in the fight against cancer. And with that, we'll leave you, our listeners, with a final thought to chew on, something stemming from this research. The source material mentions that the central nervous system usually keeps a tight lid on IFNS production, likely to avoid inflammation damaging the delicate brain tissue itself, potential neurotoxicity. So if we want to therapeutically ramp up this powerful anti-cancer axis by boosting IFNγ, or maybe its downstream signals like IL-12 and IL-15, how can we do that effectively within the CSF and leptomeninges to kill the cancer while making absolutely sure we don't cause harmful spillover inflammation or damage in the surrounding healthy brain? Designing that kind of targeted localized therapy seems like a critical next challenge, something definitely worth keeping an eye on.